what I would like to tell you today is what's happening in our lab over the last decade or so in trying to understand the neural mechanism underlying animal behavior. And I guess if I want to think, when did I start to be interested in that topic? I guess when I was a child, I was just fascinated by people's behavior and observing their behavior, and even more interestingly, listening to how they justify their behavior. And it was very clear to me that there was a huge disconnect on people's behavior and the justification that they gave afterwards, in a sense that it seemed clear to me that to a large extent, people's behavior, or my own behavior actually, follow certain route, and it's only afterwards that I start to analyze, why did I do this? Why did I choose to go to this university or this one, or follow this line of research? And so I was really interested in the idea that there are a number of behavioral or decisions that are being made in the brain, and for which the cognitive substrate of really what's happening is kind of remote and kind of resurfaces afterwards. And after my PhD, I was really interested in trying to understand, try to use new tools, molecular and genetic tools, to try to identify the circuits in the brain that organize behavior, and to try to understand what are these circuits and how do they function and how do they develop. And these with a particular emphasis on neural circuits in the brain that underlie innate behaviors. So behaviors that do not employ the most cognitive function of the brain, but are based instead, likely so, we don't really know, on some type of genetic program. And if we look at many animal species, there are really a large variety of relatively sophisticated behavior, such as the spider web waving, or building of a nest by birds, or courtship behaviors that are extremely stereotyped. They are extremely reproducible from one animal to the other within a given animal species. And animal can be raised in total isolation and still able to produce identical behaviors. And so these suggest that there is some genetic components to this behavior, and in particular that some complex form of genetic information is used to build some neural circuits that we will call hardwired, because they are present in the animal without any previous sensory experience or previous learning. And these are used to organize behavior. So if we want to understand what are these circuits and how do they function, it's important to recognize that, first of all, most of these behavior are triggered by some type of sensory stimulus or a releaser. And these are words that were defined a while back by Conrad Lorenz and Timbergen as the onset of a behavior, whether it's a somatosensory stimulus or a chemical stimulus, taste or smell, auditory stimulus or visual stimulus. And what's happening is that these stimulus then are detected by specific organ in the animal that are the sensory organs that are specialized in detecting the chemical and physical properties of the environment and translate this information into a neuronal signal, which is a train of action potential. This train of action potential is then processed through the brain and provides some neuronal encoding of this outside stimulus. And it's the neural encoding of these specific stimuli that will lead the brain both to the perception of the environment, but also to help organize specific behaviors. And so, from a molecular standpoint or genetic standpoint, this is quite interesting because one can clearly recognize that there are two parts in the system. One is the detection of the signal. What are the neurons that detect sensory signals? What are the receptors that detect those signals? And what type of constraint do they impose on then the further processing of the stimuli within the brain? And then the second part is really the coding part, the processing part. Where is the stimulus going to the brain? What are the part of the brain involved, and how can we recognize them, and how do we reach these specific behaviors? Now, our favorite sensory stimulus is a chemical stimulus, and the detection of odorants or pheromones occur in the nose, and the neuron, the sensory neuron, the olfactory the olfactory neurons in general are bipolar neurons that lie in a thick neuroepithelium, and they send a dendrite towards the lumen of the nasal cavity. And these dendrites end up by this structure called olfactory knob, in which the signal transduction machinery is expressed, in particular the receptors and all the signal transduction components that will help transduce the chemical signal into a neuronal electrical signal, which then propagate along the dendrite to the cell body, and these cells send an axon to the brain where the signal is further processed. Now, if one look at the detection of chemical signal in general, one realizes that in all animal groups, there is really a divergence or distinction between two types of olfactory stimuli. One are the stimuli that provide what we call the sense of smell, which is the cognitive recognition of specific set of molecules or volatile odorants that are detected in the nose, and then the information is processed in higher brain center and lead to cognitive reaction, memory of a smell, and a set of behavioral reaction. Now, these detections lead to cognition, and also it's a very plastic recognition, which is that according to the sensory experience of the animal, the reaction to the smell can be extremely different. And my favorite or my most vivid description of how different the reaction to a smell can be is what's happening in my lab from time to time. So, you know, I travel to France occasionally, and I bring back, obviously, some of this wonderful cheese. And as you know, the cheese that the French like the best emit this wonderful aroma. But I have to say, people in my lab don't necessarily agree with my appreciation of the smell. And in particular, I have a vivid recollection of one day where I was just enjoying a piece of cheese, and some people in my lab entered the tea room and said, "Oh, what is this horrible smell?" And you know, I was telling them this is cheese, and said, "It doesn't smell like cheese. It smells like dirty socks." And I was obviously extremely offended. And on the other hand, I had to recognize that they have a point, because one of the most pungent chemical or odorant of cheese is actually the same chemical that bring the odor of dirty socks. So obviously, I had to recognize that they have a point there. However, occasionally in the mid-afternoon or end afternoon, I'm walking through the corridor in my lab towards the tea room, and I smell this horrible odor. It's terrible. It smells like vomit. And I go into the tea room and I know what people are doing. They are eating butter popcorn. And I keep telling them, these really smell bad. And they say, what, you don't know anything. This, it's butter popcorn. Now, butter popcorn. Now, you may know that the additive, the chemical that is added to butter popcorn is butyric acid, which indeed is the main component of the smell of vomit. So I think I have a point too. And this is just to illustrate how, personal experience can really make you perceive a particular odorant in a very, very different way. So that's the sense of smell. Now, nothing like this occurs with the second type of chemical detection, which is the detection of pheromones. Pheromones are chemicals that are released by animals within a particular group, a particular species. And the purpose of this chemical is really to organize the relationship between animals, and in particular between males and females, and provide also some information about territorial defense. So the detection of chemical elicit stereotype behavior, such as mating behavior or aggressive behavior. And as we will see in a second, the model is that the detection of these chemicals lead to the stimulation of neuronal circuits in the brain that bypass cognitive areas. So these are the hardwired circuits that we are so interested about, and that I will describe more in detail now. Now, in rodents, there is a very interesting dichotomy in the olfactory system, such that the detection of odorants is provided by the function of the main olfactory system, whereas the detection of pheromones is thought to be provided by another system, the voronasal system. And these two systems are both anatomically and functionally distinct. In the main olfactory system, odorants are detected by neurons in what is called the main olfactory epithelium, or MOE, in this structure here in the posterior recess of the nasal cavity. And the information is then sent to a structure called the olfactory bulb, and then to the primary olfactory cortex, and then to a number of cognitive areas in the brain. In contrast, the dogma is, the general idea, the classical idea, is that pheromones are detected by another structure called the voronasal organ, or VNO, that is located in the septum, in the nasal septum, so in the cartilage in the middle of the nose. And these are tubular structure that contain sensory neurons, and these neurons send their projection to another area of the bulb called the accessory olfactory bulb, and these neurons in turn send projection to a structure called the voronasal amygdala. Now, the idea is that the stimulation of the main olfactory epithelium leads to the sense of smell. The stimulation of the VNO leads to pheromone responses. Now, if we look more in detail into the brain circuit, so what I've represented here is, on the right side is the circuit for olfactory detection, and in the left side, the circuit for pheromone detection. Obviously, both circuits exist in both sides of the brain. We have the olfactory epithelium projecting to the olfactory bulb, projecting to these various brain areas here that comprise the olfactory cortex. And in contrast, the voronasal organ projects to the accessory olfactory bulb that project to a very specialized area of the amygdala called the medial amygdala, which in turn project immediately to specific nuclei of the hypothalamus that are involved in reproduction and aggressive behavior. So as you can see, this circuit seems to be entirely bypassing cognitive areas of the brain. In other words, if one could talk about consciousness in animal, a rodent that has stimulation of the VNO would be entirely unaware of the perception of those pheromones and be engaged in behavior without really cognitive perception. Okay, So now, not only these two systems are anatomically separated, but they are also molecularly distinct. And just very briefly, the signal transduction in the main olfactory epithelium rely on the function of one particular channel, which is a cyclic nucleotide gated channel. So the signal transduction is based on the formation of cyclic nucleotides, and these through a G protein linked pathway. And the gating, the opening of this channel, will lead to the influx of calcium, and this in turn will help to depolarize the neurons and provide the primary electrical stimulation that will go to the brain. Now, in contrast, we discovered several years ago that neurons of the voronasal organ do not rely on cyclic nucleotide-based signal transduction, but instead rely on the function of a particular ion channel called TRPC2. TRP stands for transient receptor potential. This is a member of a gene family that now has been recognized to be essential for a lot of signal sensory detection, in particular the sense of temperature. But TRPC2 is a member of this family of ion channel, and as you will see in a second, it's very specifically expressed by neurons of the voronasal organ. And because it is so specific and so essential for the function of these neurons, we decide to generate a knockout of these genes, with the purpose of knocking down the entire neuronal circuit of the voronasal system. So here, VNO signal transduction could not occur, and therefore this entire brain circuit is silenced, if you wish. And the idea was to try to investigate the behavioral defect of an animal unable to detect pheromones. And as you will see in a second, or in a few minutes, the results were extremely surprising and led us to shatter entirely the classical idea of the detection of pheromones by the voronasal system and odorant by the main olfactory system. And this is what I would like to do today, is to walk you through the various components of the system by first explaining to you, using animal models, mutant animals, what is the global role of the voronasal system, and then identify the various components that are involved in VNO function, receptors and co-receptors, and go into the brain and up to the hypothalamus and try to identify the circuits that are involved in specific innate behavior. So this is an immunocytochemistry with an antibody against the TRPC2 protein. This is applied to a section of a voronasal organ. So you have to visualize the VNO as a sort of cylinder that is filled up with liquid. And the structure here is the neuroepithelium of the voronasal organ. So the cell body of the VNO neurons are located here, and they send a dendrite ending by an olfactory knob to the lumen, and here is where the detection of pheromone occurs. Now, again, the lumen is filled up with liquid, and the idea is that pheromones in vertebrates are non-volatile molecules, so it's very different from insects. The idea is that the pheromones are carried by the nasal mucus, and the animal, in order to detect pheromones, really need a physical contact with the source of pheromones. And if you, to help you to visualize this, just think about a dog smelling another dog. They just put their nose there, right? So that's what's happening. They are trying to get hold of whatever pheromones are around, or just smelling the sidewalk, yeah? So this is a dissected VNO. So what you would have, this would be the septum, so that's the cartilage, and the VNO would be a bilateral structure like this. So you would have these two cylinders that are right here, okay? And what we've done is we've dissected the VNO out of the septum, and we made a section like this, all right? So what you have is this long cylinder with liquid inside, and what happened is that there is an opening to the nasal cavity, And this vein here plays a very important role as a pump. So when the vein contracts, the lumen expands and suck off the liquid, the mucus out of the nasal cavity, and that brings in the pheromones in contact with the neurons. When the vein dilates, then the lumen contracts and expel all the liquid which is present here. And that's the expression of the TRPC2 protein, extremely restricted to the exact location where signal transduction occurs along the lumen. And here very beautifully on just the olfactory knob of VNO sensory neuron. So just from the expression pattern, we had a pretty good idea that this was likely to be an essential signal transduction component. And our model was that the detection of pheromones by the receptors expressed in the VNO, and I will describe those in a little while, lead to the stimulation of G protein, which in turn would lead to the gating of this TRPC2 channel. And so the idea is now to knock down this entire cascade, signaling cascade, by making a TRPC2 knockout. So we made a genetically engineered mouse line in which the TRPC2 gene is knocked down. And as you can see, indeed, on the Western blot, the expression of the TRPC2 protein occurs in the wild type, in the heterozygous, but there's nothing in the mutant. So the first question, obviously, is when you have an animal that does not have TRPC2, is it the case, indeed, that signal transduction is impaired? And in collaboration with Marcus Meister in the department, we performed a recording of the mutant epithelium using a flat electrode array. So what you have here, each of these little dots is an independent electrode, and this electrode array has 61 different electrodes. And the idea is because there are so many neurons in the VNO, and each one expresses a distinct receptor, when we have these multi-electrode arrays and we apply the neuroepithelium of the VNO, each of the electrodes can detect the activity of neurons in the vicinity, and therefore we can record from many, many neurons with just one experiment. And the design is as follows. We have the electrode array here, and the epithelium, the VNO neuroepithelium is pushed against the electrode and maintained with a mesh. And then with the stimulating pipette, we just puff pheromones. Now, you may ask, well, what are the pheromones? And I would be totally unable to answer this. We actually don't really know what the pheromones are in vertebrates, and we can talk later about why is that. We know where they are. We know they are present in urine and in sweat, in any type of bodily secretion, in tears, but we actually don't know what the respective chemicals are. And the most likely explanation is in the ability of people to purify pheromones is that pheromones are actually blends of chemical. And to have a blend that change the animal behavior, you might need 30 distinct chemicals, and people have just not been able to identify these various components. So here's our assay, and when we record from VNO from a wild-type animal, as you can see, this is the onset and the end of the stimulation with diluted urine, diluted mouse urine. As you can see, there is an increase in the firing rate of these neurons that is very reproducible, and the firing rate goes from 0 hertz to about 50 hertz. And when we do the same experiment in the TRPC2 mutant, we can never record any neuronal activity. So it seems to be the case that indeed, in the TRPC2 mutant, there is no VNO sensory activity. We know there are neurons though, and we can see them, we can characterize them. We know they are alive because we can stimulate them by applying non-specific activations such as high potassium solution. Then they start firing. But they will never fire in response to pheromone stimuli. So from a sensory standpoint, they're just silent. So the question is, what's happening to the behavior of these animals? And here was the big surprise. In contrast to everything we expected, these animals, in particular the males, are perfectly able to mate with females. Now that was not what was advertised for the function of the voronasal organ, which was one of the primary reasons we start to work on this. So here's the mating frequency of a male with a female. So if they mate, well, let's try to see the other behavior that is thought to rely on pheromone, which is aggressive behavior. And for this, we put two males in a cage, one mutant, one wild type, and try to see what was happening. And to our surprise, there was no aggression at all. But instead, the male was trying to mate with the other male. And so what happened is that we then put male and female in the same cage together with a mutant, and we tried to see what was happening, which animal did the male TRP mutant prefer. And what we saw is the animal was actually mating with equal frequency with a male and with a female. There was obviously absolutely no preference for one gender or the other of the mouse. And our conclusion from these was that the defect in TRPC2 expression in VNO signaling lead to the inability of the animal to discriminate a male from a female. Now, we thought the behavior was very striking and pointed to a very different role of the voronasal organ as what was assumed in the literature. And that really led us to a different model of the control of mating behavior, and I will come back to this a little bit later. But at the same time, we had some criticism of people who were saying, well, but you know, the type of behavioral assay that you're doing are really so simplistic. You put two mice in a cage and you see what's happening. You know, a cage is not a natural environment. So Tali Kim here, who is a behaviorist, a postdoc in my lab, decided to set up some large arenas in which she basically put the animals in an enriched environment with a lot of little toys and little things, so they can roam there at will for months at a time. And all she was doing was just observe their behavior. And I would like to show you the behavior of these animals. I think it's quite striking. In here are just wild type animal, or actually female. What you have to look at it is the left panel. Here are a group of TRPC2 males. And what's happening is that they form these mating chains in which each male is trying to mate with the male on the front and trying to be mated by the male on the back. And so that is a very robust behavior. And what we have here is a party of 3. And what we have here is a party of 5. And that was very striking because, you know, we didn't do anything. Just let them do what they wanted and observe them. And for those who know a little bit about fly genetic, also what was quite interesting is the similarity between this behavior and the behavior of a particular fly mutant, which is very well known by fly geneticists, called fruitless. Fruitless male in Drosophila are sterile, and when you put males together, they form these mating chains, exactly like what we have observed with the TRPC2 mutant. Now, the TRPC2 is a channel, right? If you knock down the channel, what you abolish is the primary event in the signal transduction. So the entire circuit is going to be silent because the initial detection of the pheromones that lead to the behavior is affected. The fruitless gene is a transcription factor. And it's a transcription factor that is expressed in many nuclei of the fly brain and is essential at the larval stage to organize, very likely, the circuit in the brain that leads to appropriate mating behavior. So there are both similarity and differences here that are quite interesting. The behavior is the same, but the defect is in one case the primary event of detection, and in the case of the fruitless, probably the entire organization of the neural network by this transcription factor. So our model emerging from the study of the TRPC2 mutant is the following. First of all, mating occurs without a functional voronasal organ. And that was totally unexpected. What that means is that all the sensory cues, olfactory cues, somatosensory cues, at this point we don't know, are sufficient to trigger mating behavior. And in contrast, there is no aggression behavior that is observed when two males are put in contact. So love, not war, if you wish. That's what's happening in the absence of VNO signaling. And the specific role of the VNO is not to trigger mating behavior, but instead to provide the animal with information about the gender of the conspecific, which in turn will lead to inhibition of mating behavior when two males are put in contact, and instead the trigger of aggressive behavior. Now, there's something quite striking here, which is that somehow the only circuit in the brain that provide the animal with information about male or femaleness of an encounter is the voronasal system. You remove it, the animal can't tell apart a male and a female. Now, I want you to think about this for a minute, because there are a male and a female mouse are different in many ways. They emit distinct pheromones, that's what we showed, but they also emit different olfactory cues. For those who've ever worked with mice, you know, when you open a cage, you can tell right away if this is a cage full of females or a cage full of males. Believe me, it's different, both in intensity and just the quality of smell. They also look different. The size of the body, the size of the head, the shape are quite different between males and females. They also emit different sounds, in particular ultrasounds. Okay? So in principle, a mouse could tell apart a male from a female using many different cues: voronasal, olfactory, visual, auditory cues. But obviously, all those cues, except the voronasal cues, are useless or are not used by the animal. So the animal can see, can smell, can hear the difference, but is not using that information at all to tell apart a male and a female, right? And what that means is that whatever other cues that are being detected are just not connected to the areas of the brain that help the animal discriminate from a behavior standpoint, okay? It's like you could see something, but somehow this visual input is not connected to the right part of the brain that would lead you to achieve a specific behavior. Only the voronasal organ somehow has organized this circuit in order to control mating behavior in function of the sex of the animal. Now, I was intrigued by this circuit, and in particular, I was very intrigued by the following question: are all animals discriminating male and female the same way, or are there specific strategies that various animal species use? And so I look in the literature, and I found, in particular in bird, a number of visual cues that are important for animals. So this is a female parakeet, and this is a male. And the animal is using exclusively this visual cue, this blue dot on the top of the beak, to recognize what is a male and what is a female. If you paint over this blue dot, then the male is now identified as a female, and other males are going to try to mate with it. If you paint a blue dot on the beak of this female, then the males are going to attack this poor female thinking it's a male. Okay, so just that dot is enough to provide the information about sex. Similarly, this American flicker has as a distinctive trait in the male this mustache. So if you mask the mustache, then this animal is recognized as a female. If you paint a mustache on the face of that female, the other animal are going to think this is a male. And Tibbegan had this very beautiful description of this by calling these the badges of masculinity. This is the sign that the animal recognizes as identifying male and femaleness. Okay? And obviously, they are both necessary and sufficient to provide information about sex. So what we found in the mouse is that these badge of masculinity are provided by pheromones, a chemical badge of masculinity, if you wish. And obviously, you all wonder what's happening in these other species. So I'm going to tell you a little bit about it, and I'm going to tell you bad news first, and then at the end of the talk, I'll give you some more better news. So the bad news is that this channel, the TRPC2 channel, is a pseudogene. It means the gene encoding this channel has many, many deleterious mutations. All these little yellow triangles here are there to indicate the presence of a deletion or frame shift or something that will just make this gene entirely unable to encode a functional protein. Now, what is interesting is to look throughout evolution, when did all these mutations occur? When did they start to be accumulated? And as you can see, they start to emerge at the split between New World monkeys and Old World monkeys. It means that Old World monkeys here, including apes, human, are totally unable to use the TRPC2 channel, and very likely so do not have a functional voronasal organ. Okay? In a sense, we are kind of natural mutants of the TRPC2 channel, if you wish, as well as all these animals. Now, what is quite interesting is that the emergence of all these mutations during evolution coincide more or less to the duplication of the red and the green opsin genes. So at the split between New World and Old World monkeys, before, in that part of the tree, these animals have just binocular vision. Bichromatic vision, which is they have two opsin genes, a blue opsin gene, and then something that is detect both red to green in just one receptor molecule, one opsin molecule. Here we have trichromatic animal. So they have three types of opsin. They have the blue, they have the red, and they have the green. And each of these opsin genes is expressed by a different photoreceptor. And therefore, the animal now can discriminate both molecularly and cellularly the presence of photons providing information about the blue color or the red color or the green color, right? So suddenly the animal has the ability to discriminate between two colors that actually have very close wavelengths of photons, whereas these animals were not able to do so. And one interesting idea is to hypothesize that these animals that now have acquired additional visual capacity require less and less olfactory capacity. And as a consequence of this, the selective pressure to keep a functional voronasal organ dropped because the animal used more and more visual cues to achieve its goal, reproduction, feeding behavior, etc. And indeed, one can observe not only in the voronasal system, but also in the olfactory system, a reduction in the number of functional olfactory-related genes in this part of the tree. Now, I have to tell you that in the voronasal organ, in human, not only TRPC2 is a pseudogene, but all the pheromone receptors that I will be telling you about in a few minutes are also pseudogenes. So there's just no way this organ is functional in human. And I have to give you a little bit of insight. We do have a voronasal organ, or at least we have a little pit, a bilateral pit here, and it's easy to recognize with a magnifying glass. But if you section through it, there's nothing that looks like a neuron. Obviously, that structure has degenerated throughout evolution. It's really just a leftover of something that used to be a sensory organ, but is not anymore. Okay? So now let's go over the players of the system And in the olfactory epithelium, the recognition of odorants is provided by the expression of a large family of genes called the olfactory receptor. And there are about 1,000 olfactory receptor genes in the mouth. And each of the receptor gene is expressed by a distinct population of olfactory neurons and leads to, therefore, the detection and discrimination of many, many distinct odorants. One considered that humans are able to discriminate between about 10,000 different odorants. Okay, and so this is thanks to those receptor genes. And in contrast, the voronasal organ uses very different type of receptors. A family of receptors called the V1R that is molecularly distinct from the olfactory receptors, and another family called the V2Rs that is also a 7 transmembrane domain receptors with a very large extracellular domain, and these domains interact with a molecule of the MHC family, a non-classical class 1 MHC family called M10. And the idea is this M10 family is a co-receptor. The M10 gene molecule is a co-receptor of the V2R and help the function of these pheromone receptors. Now, time is flying, and I'm going to skip a bunch of slides and go to the following part of the talk. So the idea is that V1R and V2R pheromone receptors are expressed in the voronasal organ, and they are actually expressed by distinct population of VNO neurons. And this information, the information provided by receptor activation, is sent to the accessory olfactory bulb, and then to the amygdala, and then to the hypothalamus. Now, what we really would like to know in the lab is what are the receptors that are associated with a specific behavior? What are the receptors that are associated with aggressive behavior? What are the ones associated with sexual discrimination? How can we identify the molecular identity of receptors providing the specificity of behavior? And we have a problem here. Actually, we have many problems. But in summary, first of all, we don't know what the pheromones are, so we don't even know what the ligands of these receptors could be. And the second problem is, for unknown reason, those receptors are just impossible to express in vitro. So if you can't set up an in vitro assay to try to identify ligand, you could imagine that you run an HPLC and get all the fraction, all the chemical fraction coming from urine, and then try to see which one will bind to a pheromone receptor. Well, we cannot do this because those receptors are impossible to manipulate in vitro. So the bottom line is we're stuck. So if we're stuck in that direction, then we thought what we could do is to try to go the other direction, which is to identify areas in the brain involved in specific behavior and try to connect those particular areas to a specific set of receptors. How do you do this? Well, using molecular and genetic tools, and starting by the identification of specific subset of neurons in the brain that are involved in specific behavior. And one set of neurons we were very interested in are neurons that release the neuropeptide called luteinizing hormone releasing hormone. It's also called GnRH for gonadotropin releasing hormone. Now, this neuropeptide is absolutely essential for the control of reproduction and fertility. These neurons, there are very few of them. It's just an amazing small number. There are about 700 neurons expressing LHRH in the rodent brain. They are located in the medial preoptic area in the hypothalamus, and they send a dendrite to the medial eminence where they release this LHRH neuropeptide into the portal vein that then provide the LHRH to neurons in the pituitary gland. And in stimulated by LHRH, these cells release LH and FSH into the blood circulation, into the blood, which is essential, both LH and FSH, into the control of gonadal development and function. So oogenesis and spermatogenesis is under the control of LH and FSH, which in turn is under control of LH. So all the reproductive function of the animal is controlled by the release of LHRH. Now, not only the function of the gonads, but in an indirect manner, the behavior of the animal also rely on LHRH as follows. The function of the gonads will release steroids, and steroids will in turn act on the brain to modulate sexual behavior, and in particular establish what is a male and what is a female brain. Moreover, the LHRH neurons also project within the brain and modulate the sexual receptivity and the sexual behavior of the animal. And the modulation in LHRH release is essential in the estrous cycle, which are the cycle of various hormones involved in reproduction and in rodents. So we thought that these neurons were really interesting as the target of information acquired by the brain that will in turn control the animal behavior. And indeed, these neurons receive a lot of information from the brain. First, and maybe most importantly, what I could call a developmental clock, which is these neurons start to function at puberty. And it's only at puberty that LHRH is released and initiate the onset of the function of the gonads. Before the puberty, the release is not achieved, and therefore the gonads are silent and underdeveloped. And nobody knows what the trigger is. It's a total mystery. Something is happening at some point, and that triggered the function of these neurons, and that's the onset of puberty. Now, these neurons are also under the control of a large variety of internal cues, because reproduction doesn't occur without having the right environment. So an animal that is stressed or starving will shut down the function of these neurons, and the animal won't reproduce. So there is a lot of internal information that is required for those neurons to function and for the animal in turn to be trigger reproduction and fertility. And finally, these neurons are modulated by sensory cues, and in particular by pheromones, chemical cues, and somatosensory stimuli. And it has been well recognized that when a male smells female urine, for example, these neurons start to increase the release of LHRH, and in turn, all this circuit is triggered. So we use a particular set of tools, which is the ability of some viruses, called pseudo-rabies viruses, to jump across synapses in a retrograde manner, so from postsynaptic to presynaptic. These viruses have been modified such that they express GFP, the green fluorescent protein. So any neuron that is infected will now be glowing fluorescent, okay? And what the virus will do is will spread from wherever you're injecting it up to neurons that are connected in and forming a circuit, right? So if we infect neurons expressing the peptide LHRH, then we should be able to jump from synapse to synapse and identify all the afferent information coming into these LHRH neurons. So in principle, we should identify all the afferent all across the brain and up to the sensory organ. We should be able to see green cells in the VNO, and those green cells should be the neurons detecting specific pheromones that are required for the modulation of LHRH neurons. Is that clear? Okay. So we've done the experiment with an additional trick, which is not only these pseudo-rabies viruses express GFP and are green, but they're also conditional. And the conditional is a genetic trick, such that these viruses will express GFP and express a gene called thymidine kinase that is essential for reproduction only if a stop codon is removed from the virus. And the stop codon is removed from the genome of the virus by an enzyme called a Cre recombinase. Now, this enzyme can be expressed under the control of the promoter of the LHRH gene. So neurons expressing LHRH will express this recombinase that in turn will excise this stop codon here. And what this does is a very neat trick, such that the virus will be able to infect exclusively the neurons that express the neuropeptide LHRH. A neighboring cell that does not express LHRH will not express the Cre recombinase, will not excise the stop codon. Therefore, the virus will just sit there and never go across the synapses. So it's a very interesting tool to identify an entire circuit connected to a molecularly identified set of neurons. Okay? And by doing this, we were able indeed to identify all the afferents in the brain that are connected to the medial perioptic area. And I'm going to skip all the control that we've done to go to the result, which is when we look in this circuit of vomeronasal system and olfactory system, where were the infected neurons? We found them in the medial perioptic area. That's expected. That's where the LHRH neurons are. We found them in the anterocortical amygdala, in the lateral cortical amygdala, in the piriform cortex, olfactory tubercle, olfactory bulb, olfactory epithelium. Forget about all those names. What is important is that all these nuclei are in the main olfactory pathway, and we didn't find anything in the vomeronasal system, nothing. Now, again, we have neurons that are supposed to play such an important role in reproduction. In every textbook that you can find on the control of reproduction, it says these LHRH neurons receive massive input from the vomeronasal system. Well, by just a very simple genetic experiment, we were able to show this is not true. Again, I'm going to skip the control, which show that although those are not infected in that experiment, they could be infected if you use a non-conditional virus. In other words, it's not because these areas are resistant somehow to the viral infection that we don't see it being green. It's because they are just not connected whatsoever. Now, if indeed there is no anatomical connection between LHRH neurons and the vomeronasal system, but a massive connection with the main olfactory system, we should have some functional correlate. In other words, if this is indeed the case, that the olfactory information is essential to control the release of LHRH, then if we take an animal that does not have any olfactory function, this LHRH function should be affected. And indeed, this is what we found. I'm going to skip a few slides. So there are two ways to obtain animals that don't have a functional sense of smell, a main olfactory system. One is a chemical lesion by injecting the animal with a chemical called diclobenil. And what you have here is a stain of the olfactory epithelium, where you can see the blue sensory neurons here and the fibers here in a control animal. And if you treat with diclobenil, in 1 week, there's absolutely no olfactory neuron anymore. They are dead. And what's very striking is this does not affect the vomeronasal organ at all. So animals like these have very specifically an impaired sense of smell, well, not the vomeronasal system. And the other way is to use mutants, exactly like we did with the TRPC2 mutant, but take an animal in which the channel that is essential for smell, that is the cyclic nucleotide gated channel, is mutated. And we use both type of animal. And what we found is that when we put a male and a male mutant, the cyclic nucleotide gated channel knockout, or an animal in which the MOE is lesion, in presence of a female, the wild type mate, but not the mutant. Not at all, nothing. Couldn't care less about the female. Same with the animal in which the olfactory epithelium is lesioned. Now, the reason we went into the trouble of doing this chemical lesion is actually because we got this animal from another laboratory who has made those cyclic nucleotide gated channel mutant. We couldn't breed them. We had no idea what was going on. We just could not breed them. Now, nobody just paid attention to this. It's just amazing that those are so difficult to breed, and the reason is they can't mate. Now, those animals have been described in the literature are not being able to detect a cookie or any type of food, but absolutely not for these type of detection that involve obviously pheromones and the initiation of behavior. So it was quite striking how severe the behavioral phenotype was, and that these mutants have been described already 10 years ago, and nobody had found that they had anything to do with mating behavior. So if I wrap up everything I told you, first of all, we found that the neurons that are essential for reproduction and fertility in the hypothalamus are connected mainly to the main olfactory pathway. And just to remind you, this fits quite well with the model we had on obtained from the analysis of the TRPC2 mutant, such that non-VNO cues are required to set up mating behavior. So in a sense, this is the anatomical circuit that correspond to the behavior that we had identified, the behavioral circuit we had identified before. And the implications of these are quite interesting. First of all, we have in our hands all the areas of the brain that are involved in the control of reproduction. And it's really the first time that anybody has described in such detail an entire neuronal circuit from the sensory organ that detect sensory stimuli up to all the various parts of the brain that are involved in setting up a specific behavior. Now, the other interesting observation is, we're not working in humans. I have no idea what's happening in humans. I know that there is no VNO, functional VNO genes in humans, but we do have an olfactory epithelium, right? And so it's quite interesting that people have been arguing back and forth whether humans were sensitive to pheromones or not. And there are a number of examples of pheromone-like effects, such as the synchrony of menstrual cycle, for example. I know it's true. I see in my lab, when there are a bunch of female students and myself in the same bay in the lab, and we work, and we all have, anyway, so it does work. And there are a number of other descriptions in literature that might be a little bit less convincing, but people have been arguing back and forth. Okay, if we're sensitive to pheromones, we need to have a functional voronasal organ. That's what was thought before. But if humans have even a little bit of that type of circuit in the brain, that would explain quite a lot some of the pheromone effects that have been observed just through the main sense of smell. So that's good news for the perfume industry, I guess. And obviously what we are going to pursue in the lab is the investigation of the second part of the circuit, which is what are the areas in the brain that do this, the sex discrimination, what are the areas of the brain, what is the circuit, and how are the two systems interacting to help the animal perform the appropriate behavior. And I will finish here and thank people in the lab who've performed the work. Thank you very much.